So, so today's lecture is on therapeutics and resistance, and this lecture is based on the assumption that the graduate students already learned about all the microbes and how they cause diseases. And then we'll also address how they become resistant to many of the antimicrobials we have. And first of all, even though you should know what microbes are, and just a quick, like a one-page review of what microorganisms are, and they are critical part of our human system, and we can live without them, but we can't live with them, and with some of them, but we cannot live without them. They can be unicellular or multicellular. When we talk about unicellular, we're talking about bacteria. The proper name for bacteria is eubacteria, since we discovered archaeobacteria, which used to be part of bacterial kingdom. And then the viruses that require hosts, the fungi, the unicellular fungi is your yeast or candida. And then of course the unicellular eukaryotic organisms such as amoeba, giardia, and plasmodium and leishmania species. And the multicellular organisms that cause diseases, obviously we're interested in, is fungi and metazoa. A metazoa example is your parasitic helminths or worms. So we literally going to run through this whole thing and in an hour and a half. So therapeutics, I want to make sure that all of you have a very good understanding of what therapeutics are. And when somebody talks about antimicrobials, they're talking about anything against the microbes, that is your eubacteria, virus, fungi, and parasites. When you talk about antibiotics, it's often against the eubacteria. Antivirals are against viruses, antifungals are against fungi, antiparasitics are against the parasites, and if you say antihelminthic, they're specifically anti against helminths or worms. With the problem of antibiotic resistance, which we'll discuss in a little bit, there were a lot of efforts to repurpose some of the drugs. The question is, can we use some of the antivirals against other pathogens or fungi or parasite? And we found that a couple of antifungals and antibiotics can be used against parasites. Examples are rifampin, azithromycin, both are antibiotics, amphotericin and metronidazole are mostly used for antifungals. The mode of action could be topical, mode of use rather, you can use it as wipe on your skin, this topical use, oral, you can take as a pill or liquid, or intramuscular, where you inject through your muscles, either dorsogluteal, which is your butt, or deltoid, which is your shoulder muscles. Those are the two preferred because the skin is quite thin in this area, and then you inject your antibiotics through them. You can get intravenous, so through your blood, or intraperitoneal, you just lift up your stomach and stick it slightly under the peritoneum, and that's how the drug is administered. So once, if you're a doctor, you should know which is the best form to administer certain drugs. Okay. So the question is how one goes about looking for drugs. Obviously, so drugs are targeting bacteria, virus, and pathogens, right? Essentially. So how would you go, what would you choose to target? Joseph. And I'm going to Florida next, and I'm going down the list. So think about it.
I'm not sure what you mean specifically, but I guess you'd want to target something that is not gonna heavily impact the host, but should be specific to the target in question. So I mean, maybe if you're dealing with gram-negative bacteria, you want something that'll target LPSs.
Okay, that's a good answer. That is probably encompasses most of it. Give me an example of a target, Florida. besides LPS? You already gave me an LPS, though we don't have any antibiotics that target LPS. So in fact,
I
there's
know
none.
some antiretroviral drugs, they target the binding site where the virus enters the cell. They can target the where it replicates and things like that.
Right. So you went for what you know, which is HIV. Florida, you need to expand your answers. Okay. Yes, you need to know HIV because that's your area of research. I like to see more of broader understanding of antimicrobials. Thank you for the answer. Christian, any thoughts? I know this is new to you.
Possibly one would like to target receptors on molecular, the membranes on specific organisms, if
That's
they're
a good
unicellular.
answer. Yeah, you can prevent the receptors from attaching to the virus or attaching to fungi, unicellular fungi. That's a good answer. I'm going to skip the other two. I'll wait for the next questions. Okay, so what are the ideal antimicrobial agents or antimicrobial? It's something that's very soluble in body fluids, right? It doesn't precipitate. It should be stable. It should be degraded and excreted by the body slowly. That means you have longer, longer half-life as well as longer shelf life. Spectrum activity, preferably an antimicrobial that targets more than one organism, which we call the broad spectrum. On the other hand, we also would prefer a narrow spectrum targeting a single organism. We have to be concerned about toxicity, that when a person eats this drug or takes this drug, has very little or no side effects, that it doesn't interact with any food. Of course, many of us like to drink alcohol. We have to make sure these drugs don't interfere alcohol. And then some drugs, as you know, can cause allergy. For example, many people have penicillin allergy. So you have to make sure the drug is non-allergic. And if you're a doctor, you always determine whether the particular person is allergic to any known drugs before administering a drug. Uh, and of course, the most important thing is it should be at a reasonable cost so most people can afford it. So, and like what Joseph said, you want to focus what is unique to a microbe, which is often is the cell wall is extremely unique. It's not necessarily the LPS, because lipopolysaccharides is still uh, a membrane made up of lipids, right, which is very similar to our cell membrane. So we do not want to give anything that would also affect cell membrane. But however, the cell wall, the peptidoglycan, is extremely unique to bacteria. So as you can imagine, many antibiotics target the cell wall, and not just the cell wall per se, but any enzymes involved in synthesis of the cell wall. Fungus have many unique cell wall components, and you can certainly get it. And then we, of course, always want to target some novel gene products that say the bacteria makes a toxin, for example, cholera toxin. Only Vibrio cholera makes it, so maybe we can target something against cholera toxin. And then the other important thing is that whatever we focus on of a product that against a pathogen, it has to be part of the core genome. And this concept of core genome is applicable to many bacteria. We probably true for other microbes, but there's not much work out there. But in terms of microbes, what we learn is that bacteria particularly is extremely versatile in collecting DNA from the environment in order to make itself stronger, or to fight against other pathogens, or to fight against antibiotics. So they constantly acquire genes by what we call horizontal gene transfer, means it's taking genes from different strains, it's not vertical, but once it gets into horizontally, then it can pass it vertically to its daughter cells. So if you make an antibiotic that is part of what we call the accessory genome, the genome it acquired. And what happens to the bacteria is that it says, oh, you are targeting my accessory gene. I'm going to kick it out. I don't need it. I don't need to survive. So they kick it out. So if you focus on something that the bacteria absolutely needs to survive, then that's a better target. And of course, we don't want any side effects, and we do want to make sure that there is no human counterpart. And again, it's preferentially found only in the pathogens. And the main reason is that, what do you think, the last reason, Sophia? Why we need to make sure it's only found in the pathogens?
That way you don't hurt the host.
Yeah, but you don't hurt the host. That's the main thing, right? You don't hurt the host system at all. There's another answer, but I'll come to that later for that question. All right. So when we talk about antibiotics, I told you it's against bacteria. There is an antibiotics database, and you can get a very quick overview of what antibiotics to use. Go to, if you link to med bullets, it's commonly used by medical students. But if you want to get an idea what antibiotics to use, that's a good place. Of course, if somebody asks you, especially if you're a PhD student, you soon will have your parents asking you, oh, you are a studying PhD, tell me what is this antibiotics all about? Suddenly you become this a pseudo doctor to your family if you're the first PhD, right? And I'm a pseudo doctor to my family members all the time, right? So there is a Merck manual, which I did not give you the link to. If you just type up Merck, MERCK, on all antibiotics and what you use it against. And of course, a Merck manual is constantly updated, and CDC also updates the antibiotics and options. And then this also then, we know a lot about antibiotic resistance. We can certainly would have a database on antibiotic resistance. All right, so let's get to antibiotics. So there are classes of antibiotics. They are based on its structure. You divide the antibiotics, they are called beta-lactams, which is the largest class, macrolides. Some of them, we'll see all those classes shortly. And you can also divide the antibiotics based on how they act, bactericidal versus bacteriostatic. Amrita, do you know the difference between bactericidal and bacteriostatic?
Yes, bactericidal will kill the bacterium, bacteriostatic will inhibit its growth.
Thank you. So bactericidal is, of course, hang on, you guys can hear me? So bactericidal is it immediately kills the bacteria. Bacteriostatic is the bacteria will stop dividing, and as soon as the antibiotic gets washed away, then they start dividing. And this is very true for beta-lactams. And then you can also divide the antibiotics as narrow spectrum or broad spectrum. When they say broad spectrum antibiotic, it usually attacks both gram-negative and gram-positive. I assume all of you know what's gram-negative and gram-positive. Perhaps Christian doesn't. Christian, make sure you go read about it and make sure it gets into your head forever, right? What's the difference between gram-negative and gram-positive? Very quickly, the difference between gram-negative and gram-positive is what we have on the membrane. Gram-positive as lipothiacoic acid, whereas gram-negative as O-antigen lipid A. Gram-positive has no outer membrane, gram-negative has two membranes, inner and outer, and gram-positive has a thick peptidoglycan layer right here, and then gram-negative has a very thin peptidoglycan layer. And all this has an impact on how we treat them, and understand the physiology of these organisms. And another main thing that happens is, this is again your gram-positive, your gram-negative mycobacterium, right? Mycobacterium tuberculosis that causes TB has a completely different cell wall. The inner membrane is pretty much the same. They also have a peptidoglycan, but they have arabinogalactan and mycolic acid that makes it extremely thick. Hence, it's why it's very hard to treat TB. And as I told you, cell walls of these bacteria are very different from human cell walls. So many antibiotics to date has targeted cell walls. Beta-lactams, they bind to penicillin binding proteins. These are proteins and enzymes that are responsible for synthesis of the peptidoglycan. So if you prevent the synthesis, then the cell wall cannot form, and then eventually, so what the bacteria does is just stop doing anything, right? But if it is dividing, it will kill the cells. But if it stop dividing, then it won't do anything. So that's why they are bacteriostatic. And then vancomycin and cycloserine, they inhibit cross-linkage of peptidoglycan layers. So peptidoglycan biosynthesis is extremely complex. We can talk about it if you're interested some other time, because my lab worked a lot in this area. And then bacitracin is, it inhibits the inner membrane or the cytoplasmic membrane. And this basically prevents the peptidoglycan precursor moving into the peptidoglycan synthetic pathway. And we talked about mycobacterium. They have a mycolic acid synthesis. So we have a special drugs, isoniazid and ethionamide. They both inhibit mycolic synthesis. And then, of course, ethambutol inhibits arabinogalactan synthesis. Because this cell wall of mycobacterium is so thick and it's very hard to control Mycobacterium tuberculosis, you actually get treated with all three drugs all at the same time: isoniazid, ethionamide, ethambutol, as well as rifampicin, if you will talk about it shortly. So the major target sites is, of course, number one is your cell walls, just we talked about. However, you can also target replication if there is special enzymes that are used for replication, especially true for virus, because the viral RNA polymerase are viral encoded, and you can target the viral RNA replication. You can target transcription because the enzymes that used to transcribe RNA polymerase is very different for parasites and is very different for bacteria than hosts. So you can certainly target the process transcription, particularly RNA polymerase. And then going from RNA to protein, I hope you guys remember your molecular biology. When RNA to protein, that process is translation. It requires ribosomal RNA or ribosomes. Our ribosomes for bacteria, 16S and 23S and 5S ribosomes, as well as for fungi is 18S and 25S, and I think it's 5.3S. So the ribosomes are very different for all the pathogens. So targeting the ribosomes is also very one way to get rid of pathogens. And then you go after specific activity of a protein. The protein could be RNA polymerase, for example, right? You can go after a protein. You can go after penicillin binding protein I just talked about. There are other gyrase, for example, right? So we can go after specific protein that can affect any of this process. Or you can understand a metabolic pathway. One of the things is that if you find that this particular pathogen you're working with has a very unique pathway to make folic acid, for example, and we depend on folic acid from bacteria, and we can target some of the folic acid biosynthetic pathway of a pathogen. And again, it's there are antibiotics that targets folic acid metabolism. So here is a list of antibiotics that I just talked about, and here is your replication. It targets DNA gyrase, and the antibiotics is quinolone. Cell wall synthesis, beta-lactams for gram-negative and glycopeptides as well, mostly for gram-positive. Folic acid metabolism, we use sulfonamide or trimethoprim. And for protein synthesis, we have quite a few. There is macrolides, example of and chloramphenicol and clindamycin, all different classes. And there's also aminoglycoside and tetracycline targets separate components of ribosomes, 50S, 30S. And then you have an antibiotic that do target the inner membrane, namely the polymyxin. And this is probably our last resort drug, and we don't use it a lot, and we'll come back to. And then we have the DNA-directed RNA polymerase, and we have the rifampicin that targets the RNA polymerase. It's also sometimes known as rifampin. Rifampicin is the same. So here is the examples of antibiotics, and these are the various classes. And the beta-lactams, I'm going to come to it in a second. Many of them, like this, in the case of tetracycline is very toxic. So we use doxycycline, and this is new one, minocycline. Pretty commonly, you hear people taking erythromycin and azithromycin or clarithromycin. Again, it all depends on if your antibiotic resistance profile. And there's aminoglycosides, these are gentamicin, tobramycin, and amikacin, and so forth. And so you can see there are cyclic peptides, lincosamides, clindamycin is a lincosamide, and then this oxazolidinone, this linezolid, and then of course there are antibiotics with sulfur in it, so they're known as the sulfur antibiotics or sulfur drugs. There are people allergic to sulfur compounds, so you need to make sure they're not have sulfur allergy. So the beta-lactams, when people discovered that bacterial cell wall is unique, but that didn't really result in the identification of the first antibiotic. We don't have a lot of time to go through the history, but many of you probably know the story of Alexander Fleming, who was lazy to clean, and he left his plates on his bench and went on a Christmas break and came back and found as he was going to throw those plates away, and then he saw zone of clearance, and he saw something in the middle, which now we know is a fungi, and the bacteria's growth was inhibited. And it took another 20 plus years for his students to purify penicillin. And then once we discovered penicillin, and then as the bacteria became resistant to penicillin, they started, once you know the chemical structure, the scientists tried to modify the side chain, and they tried to make new antibiotics that are more potent than penicillin, because within a few years, penicillin resistance became rampant. And then, of course, penicillin, ampicillin, all three are very similar in activity. Methicillin is mainly for Staphylococcus. And you guys know about MRSA, there's methicillin-resistant Staphylococcus aureus. And then now oxacillin, and we go down, all the cillins are down here, right? And then they discovered another group of naturally produced beta-lactams. Those are known as cephalosporins. And then the first generation of cephalosporins, and again, for the same reason, as the bacteria become resistant to cephalosporin first generation, they create the second generation, third generation, and now we are on to fifth generation, because many bacteria become resistant to a first generation, but still some of these are quite used in this country. And then we also started synthesizing what we call carbapenems. And these all have penem in the end. There's only 5 of them, quite commonly used. They are broad spectrum antibiotics. And then the monobactams, Astronam is used most often. The others are quite new, and we haven't quite seen the use. Astronam is still very much in use. And here, I'm not expecting to memorize anyway. Graduate students know this is a lot of them for knowledge, and you can see the target of these various beta-lactams varies, right? So here's most of the cephalosporins are broad spectrum because they cover gram-negative and gram-positive. You keep some third generation for serious infections as meningitis and pseudomonas and so forth. And when you use monobactums, when your bacteria is resistant to beta-lactamases, right? It should be resistant to beta-lactams. So this is probably about the most commonly used antibiotics are beta-lactams. It's very hard to cover toxicities without going into individual drugs. So here is just a sample of the toxicities we see. There is, hypersensitive reaction to penicillin and methicillin. Vancomycin, we don't use it until the patient has MRSA. And the reason because it has nephrotoxicity, there's a kidney problem, ototoxicity, there is your hearing, thrombophlebitis, which has this make you diffuse flushing, right? Affect your blood. And however, it does not cause many problems. And then aminoglycosides, you have ototoxicity, renal is kidney problem, and it can also cause neuromuscular paralysis to some people. So I don't want to go through the whole detail, but basically you need to know that almost all antibiotics have side effects. You always take the the word I'm looking for is the gain versus loss, right? That's a different two words you can use. It will come back to me later. So whether if treating them is going to protect them or kill them, right? If you don't cost benefits, pros and cost to benefit, right? It's a cost benefit ratio basically, but that is in so risk versus benefits. That's what I'm looking for, right? Risk versus benefits. Either risk worth taking for saving somebody's life. So we're looking for risk versus benefit. And that's a decision every medical doctor has to make. All right. So then, as you can imagine, like I told you, bacteria fights back. It does not want to die. And the first thing it wants to say, no, I'm going to create this my membrane such that it won't let any antibiotics enter. And Pseudomonas aeruginosa is a good example. Its membrane is 6 times more impermeable as compared to E. coli. And then if the drugs do get inside, they all have pumps that will pump it out. We call them efflux pumps, that is effluxing out. And we'll see a couple of examples later. Or you can do what you call a target modification, right? So you modify its target. If the gyrase isn't necessary, where these antibiotics bind, it will change the amino acid. So now the antibiotics cannot bind. Same thing can happen to ribosomes. It will change the nucleotide. And this is a gyrase, of course, as you can see, is opening up the DNA. And then it can also generate enzymes to inactivate the drug once it enters, or it can inactivate the drug while on the membrane or inside the periplasms. So it can have many ways. So it can restrict access, promote exit, alter the target, or inactivate. And some antibiotics need to be activated inside the cell. When it gets activated inside the cell, the bacteria can say, no, I'm not going to activate these antibiotics. I'm going to just turn off the enzyme. And in extreme cases, particularly is quite common in the gram-negative, they lose the cell wall. Can you imagine a bacteria without a cell wall? It happens. They simply stop making any peptidoglycan. So cell wall provides the structure, right? The rod-shaped bacteria, in the absence of cell wall, it becomes round. But once the antibiotic is gone, they will make their cell walls, and these called the L forms. And then they can, the other main thing they do is, like I told you, they acquire resistant genes, and that is the biggest problem, more than all the others.
Wait, they can survive with other cell walls?
Yep.
Oh, wow.
So
I thought for some reason they would just die as soon as their cell walls collapsed.
no, And the absence of cell wall was discovered very, 50 years ago, but people know it, and now they're beginning to acknowledge that, and they can live, survive without the cell wall, because it's double membrane, so the cytoplasmic content is still protected, right? Cell wall provides the structure and resistance. So as you can imagine, when we're talking about resistance, it's going to become resistance to all the points where you're attacking the antibiotics. So that's all the target alterations. So I want to just talk about one or two examples just to give an idea. And I told you that particularly beta-lactams that are targeting cell wall. And one of the things that very obvious about beta-lactam antibiotics is the presence of this ring called the beta-lactam ring. Okay, this box square here, that is the beta-lactam ring. And as soon as we started treating the bacteria with the beta-lactams, they started producing enzymes that actually will cleave this beta-lactam ring. So this enzyme is known as beta-lactamase, and there are many beta-lactamases, and they're specific for beta-lactams except for monobactams. Remember the last one, the monobactam? It has a single bactam ring, doesn't have this double ring. And so they are usually secreted into the periplasm, these enzymes. And of course, gram-positive secretes it into the extracellular fluid because it doesn't have an outer membrane. And as we introduce antibiotics, there were constantly new beta-lactamases emerging in the environment. Right? So remember, we talked about 4 classes of antibiotics, beta-lactams, right? The penicillins, the cephalosporins, the carbapenems, and the monobactams. And we don't have an beta-lactamase for the monobactams, but however, we do have 4 classes of beta-lactamases. It's the same slide. Okay, and so because they were producing beta-lactamases and we still wanted to use the antibiotic we had against beta-lactams. So what they did is they then looked for specifically inhibitors of this enzyme called beta-lactamase. And you probably would heard of clavulanate because it can be given ampicillin with the clavulanate. And in fact, there are 3, there is a new one, avibactam is a new one. I don't have a picture of sulbactam. Okay, so the clavulanate, this is clavulanic acid, sulbactam, tazobactam, and avibactam are beta-lactamase inhibitors. So if they go in and administer them in parallel with ampicillin clavulanate combined. So the beta-lactamase inhibitors will prevent the beta-lactamase so that the antibiotics can act on the cell wall or on their targets. So these are a couple of combinations available in the market that gets used because when they know that this bacteria is beta-lactam resistant. And then gram-positives, so of course, particularly staphylococcus, they have a different issues. They don't have an outer membrane, but they have, we have this vancomycin, right? And what happens is vancomycin basically binds to this D-ALA side chain of the peptidoglycan. And so this is how vancomycin works on gram-positives. So what does Staphylococcus do? It changes the D-ALA to D-ALA lactate. Now, vancomycin cannot bind. This is how they become resistant. Right? And this is by acquiring some genes. We don't know where they got the genes from. And this is what happens when the D-ALA mutates. So an MRSA and a VISA, all those are strains that have mutation in the cell wall. And in fact, this is a very quick summary. They introduced in 1940, by 1941, all of them became resistant to penicillin. And then they introduced methicillin, and then they become MRSA in the 1990s, right? And then they introduced oxacillin. Now we have ORSA. It's not very common still, but MRSA is very common. And then they also have intermediate susceptibility, visa and GISA, right? So, but you probably hear a lot of MRSA in the hospitals.
Can't they do rotations though on like the medications that they're using, like every 10 years or something, to like target something they should have lost?
They should.
Like if you don't use penicillin for 10 years, won't penicillin become viable again?
It is true, and they are testing that, but it's very hard to institute. It is recommended not to use penicillin, but if you go to third world countries, you can't stop it, right? And the drug companies are happy to dump. So it's a battle, and it's an ongoing battle. Yes, you should rotate the antibiotics so that and we don't know how long it takes, but usually in the lab, a bacteria can lose the marker very quickly, but in the wild, in the environment, we'll talk about a little bit, because the problem comes in multifold, right? So I have a slide on that to that effect. And here is the efflux pumps, and you can see one of the problem with the efflux pumps is the fact that these pumps, gram positive also has a efflux pump, okay? And this is example given is a gram negative, because as you can see, it's a double membrane. There are 5 different kinds of pumps. And the problem with these efflux pumps, and I still remember when it was first discovered, I was a graduate student. And what we learned is that they are non-discriminatory, which means that they simply pump any drug that comes in. And you can see this particular one, the SMR complex, or the ABC transporter. You can see this particular, look at how many different drugs, classes of drugs it's pumping. So if a bacteria acquires this gene, NORA protein, then that's it. You can't use any of these drugs. And and imagine this is only a single protein, a single gene. You can acquire a single gene and pump it out. And these all
Are all these pumps present in the same bacteria, or is each pump specific to a species of bacteria?
So each pump is specific to a particular bacteria, but there will be similar pumps in other bacteria. And the thing is, there's not only one pump in one bacteria species. Like Pseudomonas, I believe 7 pumps, right? When you have that many pumps, it's 5 to 7 pumps at least, right? And you basically, it becomes resistance to everything, right? And this is a little bit complex. You need 4 different genes, and this is called R&D pump. Is a terrible name because the original protein was involved in nodulation, and now that's the D stands for. Anyhow, so these are different. The names come from original protein, right? When they first identify the first protein involved in a particular pathway, and then you name it. All right, so that's for the efflux pumps. So let's just then understand a couple of resistance terms, right? And we have what we call the multi-drug resistant, what we call them the superbugs. That means they're resistant to many drugs, but there are still quite a few drugs that you can still use it to treat these bacteria. And then there's a second group, which is extensive or extremely drug resistance, and they're known as the XDRs. And these are resistant to one antibiotic in each class and susceptible to 1 or 2 antimicrobial categories, right? Maybe, beta-lactams, I can't use penicillins, but I could use something else, a monobactam. However, pan-drug resistance is the PDRs. It's literally resistant to most antibiotics. Maybe there's 1 or 2 drugs. And the 1 or 2 drugs these days are either polymyxins, mostly it's a polymyxins that you can use. And if it becomes completely drug resistant, there's nothing we can use. So because of these issues, the CDC has divided the pathogens into 3 groups: highest priority, second highest priority, and third highest priority, which is emerging pathogens. And basically, if there are bacteria in the highest priority, that means we need to find new drugs for them. And WHO has a somewhat different classification, but addresses the same issue: critical pathogens, high importance and medium importance. And that's basically, if they're critical pathogens, you need antibiotics badly. They are very likely a pine drug resistance or XDRs, PDRs. And then we have what we call last resort antibiotics. These are the antibiotics that you are not supposed to use unless it's absolutely necessary. It's a life and death matter. And this is called the colistin. And the example is a polymyxin, that's a colistin, right? So what contributes to resistance? And this is, I really want guys to understand. Hi, Fatuma. Did she just come? Okay. All right. So one of the reasons for resistance is overprescription of antibiotics. And this overprescription happens because people, it's not just the doctor's fault, it's also the patient's fault. You have a common cold and you go to the doctor and the doctor says, okay, here, you take ampicillin. You feel great because, oh, now I got an antibiotic, so I'm going to feel better. But that antibiotic doesn't work for your cold. So what happens? Now you are exposing your bacteria to these antibiotics. And then they slowly will become resistant. And the other reason is when somebody gives you a prescription, you never complete your prescription, right? And you say, oh, I feel better, now I should stop eating it. But what have you done? When you take antibiotics, you kill the first layer of bacteria, and the second layer is sort of sensitive, but it's still there. If you don't come back with your second round of antibiotics to kill them, now they will develop resistance. So you must finish your course of treatment. And then the other reason is also overuse of antibiotics in livestock and fish farming. All farmers want healthy animals. They want to make a lot of meat, either chicken or beef or lamb, it doesn't matter. So there's a lot of fish, for example, and they use a lot of antibiotics in their feed. So what happens when they poop, and all the poop has antibiotic resistant bacteria, and then some of the poops gets used as manure, and now the manure has your vegetables have manure with antibiotic resistant bacteria, and then now you get exposed to this antibiotic resistant bacteria. And then there's also poor infection control in hospitals and clinics. Hospitals and clinics are the worst places to be. If you're sick, you think you're going to get better in the hospital, but the hospital has the most resistant bacteria circulating in the hospital. So unless you absolutely need to be hospitalized, I would say don't go, right? And now we know that with COVID-19, they say don't go there unless you absolutely need breathing support. Then lack of hygiene and poor sanitation is now, with the COVID-19, everybody talks about hygiene, washing hands and cleaning your hands after you poop and using the restrooms. And then poor sanitation is a major problem in developing countries or third world countries. And that's where a lot of effort is gone in building sanitation facilities, especially in Africa and some parts of Asia. And also South America, right? So we have a lot of poor communities and in rural communities. I remember in 1993, I was in France and we went to visit somebody in Brittany. France is this very advanced country. And when I asked for a restroom, he said you have to go to the apple orchard because they didn't have sanitation facilities. So that was something, right? Okay, so and then the other major problem now is lack of new antibiotics being developed, because the antibiotic pipeline from the day they start identifying synthesis and get to market, it takes to 10 to 15 years. at least about $10 billion to go from A to B. And so most of companies has gotten away from it because now they become resistant, and all the money they spent on making these antibiotics now no longer useful. So that's a huge issues with a lack of development. And they now want the professors in research labs to come up with new antibiotics, or at least potential targets. So we talked about this livestock use, and in fact, 70% of the antibiotics in the world is consumed by animals, and only 30% consumed by humans. So the husbandry, there's a major need to convince the farmers and all those people to use no antibiotics or less antibiotics. You guys can ask me a question later about the antibiotic-free meat. I can tell you more about that later. So 1 minute break. That's already 50 minutes into the session. We only have another 40 minutes for covering antivirals and antifungals and antiparasites. All right, just take a minute break, guys. All right, I'm back. So let's talk about antivirals. And the important thing is, virus control is a little bit harder. The best is to disinfect, immunize, quarantine, all of which is happening right now with this COVID-19, and elimination of the vector source, right? And important is educating the public about the virus control. And as far as viral antivirals go, and disinfection depends on the virus, whether it's enveloped or whether they have only capsid. And most of the time you need to autoclave in order to kill the viruses. And you can use bleach solution or soap or solvents. And as I told you, I'm going to go a little bit fast. So the best way is to immunize. And we know that very well now with COVID-19. We are trying to immunize everybody in this country and in the world. And this is how you would control the spread of the virus. It was very well known before COVID-19. And then, of course, quarantine is to limit the exposure. And in many cases, we eliminate the vector. And unfortunately, there is no real vector in the case of SARS-CoV-2. it's goes the transmission is through human to human, and we need to figure out how to control potential source of the viruses or parasites. We, one of the, with the virus is also there are viruses that cause sexually transmitted diseases or transmitted sexually, for example, HIV. And then there is human papilloma virus, HPV. We need to then educate people on safe sex. Oh, it's coming again. All right. So this basically what we just talked about. So in terms of immunization, many of our immune vaccines, they what we call active immunization. You provide the live virus or attenuated virus or inactivated virus, and these are all these various vaccines that's available. And the thing with this is it provides you long-lasting immunity. And then you can also have a temporary immunity, which is using passive immunization. You probably heard of convalescent plasma for COVID-19, or you can have antitoxins. This is often either if you haven't had a vaccine and you have to go to a meningitis belt area like Morocco, and he says, okay, I need to get a shot of immunoglobulins before I leave for the country. And so there are a couple of immunoglobulins available for prophylactic treatment. For example, rabies. If you get bitten by a dog, you already introduced the virus, so immediately you get the immunoglobulins to prevent the further spread. But it is temporary immunity. You must get the vaccine for them for a lifelong immunity. Unlike bacteria, we have much fewer drugs for viruses, but unfortunately, they're also more toxic. We have made great progresses for herpes, HIV, respiratory, and hepatitis virus. Again, preventive vaccination is the key. And what Florida said, a lot of viruses, are preventing the entry, so that focuses on the receptors. When the virus enters, it often has to uncoat to release the genome, and you have antivirals that focuses on uncoating, particularly amantadine and rimantadine in flu virus. And then you have against nucleic acid synthesis, and then as prevent the assembly, as well as exiting and attaching again. So there are many different drugs, and here as because of HIV and as such a huge problem, there are all kinds of inhibitors, from fusion to binding to fusion to synthesis to gene expression to assembly and maturation. So you have all kinds of various antiviral drugs, and often resistance comes from using a single drug. So you use multiple drug, which is HAART treatment, which is a multi-drug. Very quickly, you will hear about these drugs quite a bit in your life. This is acyclovir or cidofovir or famiclovir or ganciclovir. And all these are guanosine analogs that targets viral polymerase. And the difference is some of them requires a phosphorylation for activation, some not. So since when it requires phosphorylation, many of the viruses becomes resistant because they need the host polymerase, the bacterial polymerase, and they will become resistant, right? So they came up with a cidofovir, which doesn't need that. And I briefly talked about amantadine and rimantadine. And if you know the viral structure, they have two major proteins on the surface. That is the hemagglutinin, these blue guys, and then the red guys are neuraminidase, and then this is the ion channel, M2 ion channel. And so the amantadine and rimantadine blocks this M2 channel, and then this oseltamivir and zanamivir inhibits the neuraminidase, right? And then the ribavirin is a guanosine analog. So knowing the structure, knowing how it looks like, what proteins found on the surface, and you can target drugs against them. However, many of these drugs don't prevent infection, right? But once it enters, it can slow down the progress. The weapons of choice, again, there's many options, and most of it, you can see the guanosine analogs is the major option for many of them. And these are the various viruses with the particular antivirals that are available. And I want to focus on one of this IFN, and I'm going to talk about a little bit of just very quickly. These are the various guanosine analogs, and this is, the use can change, right? You need to know whether how to use it. And ganciclovir is intraocular, where you just put a drop in the eye. So it changes. Again, they all have a lot of toxicities, as I told you earlier. And same thing with herpes, the cold sore is herpes, correct? So you can use topical cream or you can take drugs. Again, you can see the toxicities. There's a lot of ocular toxicities or renal toxicities, which are not very good. One of the things I just want to make sure you guys know about this is the interferon, right? There are interferon as a drug. If you've done the MCB, you know interferon is very much part of a host immune response. It's a cytokine that is responsible to tell the host cell that it's being infected. And so the interferon is basically induces the host to make enzyme called RNase L that can hydrolyze the RNA genome, and it basically induces antiviral state, right? So it's a very important cytokine, and there are 3 types of it. And what happens is people said, okay, maybe if there is a viral infection, we give them before even the body starts making it, takes time to make. What if we give the interferon and then the body can react much faster? And so they have interferons against papilloma, Hep B and Hep C. And sometimes it's get pegylated. You see pegylated interferon as your drug, and that is basically to increase its half-life. This is simply summarizes what I just said. There are, this is a specific weapons of choice for various infections, and the pegylated interferon for Hep B and Hep C and others. All right, and then that's a quick run through with antivirals. Okay, so there's a very few drugs, right? So interferon, agonisine analogs, and then there's amantadine and rimantadine. Of course, there are a lot of drugs being developed against SARS-CoV-2 now, but nothing in use. So fungi, antifungals, and as you remember, if you know, you should know that the fungal has different structures. They have spores, hyphae, rhizoid, cell membrane, and cell wall. They could also have capsule if they're unicellular. Important thing is the fungal cell wall is very different from human cell wall, bacterial cell wall, a parasitic cell wall. They may share some stuff with the parasitic cell wall. They're very rigid. They have these polysaccharides, chitin, glucans, and mannans. If you remember, chitin is also found on insects, right? And then they also produce melanin, and melanin, that's what makes the black fungi. When you have a fungi on your bread, it turns black, and that's due to melanin production. And so as you can imagine, once we know those cell walls, we figure out how to attack the cell walls because they are unique. And we also figure out how to, if it also focus on the synthetic pathway. One of the major cell wall component of fungi is ergosterol, right? It is also found in protozoa, particularly trypanosomes. And this is equivalent to cholesterol, but it's not cholesterol. It's somewhat similar. And so you can imagine the ergosterol has become a major drug target, and there's amphotericin B, nystatin, or you can have ketoconazoles. And you probably, if you have ever had a fungal infections, you've probably been given these creams to prevent or to protect, and mostly usually it's topical. And many of these have very severe toxicity. So as I told you, some fungi has capsules such as Cryptococcus neoformans. This particular fungi is very important in the case of cystic HIV patients when their T cells really drop quite low and they get these Cryptococcus neoformans and which can kill the patient. We call this cryptococcosis. And so you could have drugs that target the capsule. So here is the 4 main antifungals that is available. As I told you, you go after ergosterol, and we know the ergosterol pathway goes from squalene to squalene epoxide to lanestrol to ergosterol. So you have antibiotics that particularly target this squalene epoxidase, so it can't make squalene epoxide. I have something is missing here. Okay, so I think this is echinocandin, I believe. Maybe the next slide I'll see it. And then you can also prevent ergosterol per se, and this is done by making holes. You can prevent the nucleic acid synthesis with the analogs, or you can prevent the cell wall synthesis. So let's see if I have the list of those drugs. There you go. Right. Okay. So here you have all these drugs focus on ergosterol. Terbinafine, right, attacks squalene epoxide. So going from lanestrol to ergosterol, there is an enzyme called the 1,4-alpha demethylase, and all the azoles, the fluconazole, ketoconazole, itraconazole, clotrimazole, they all attack this 1,4-alpha demethylase. And then the ergosterol in specifically, these are the polyenes, and this is amphotericin B and nystatin. Nucleoside analogs, this fucocytosine is only one, and then the cell wall synthase, you have echinocandins, and there are 3 of them: capsofungin, micafungin, and anudilafungin. So these are the summary of the antifungals. And the polyenes, such as the amphotericin, basically, attacks this ergosterol so that it causes the potassium to come out of the cytoplasm and causing the cell death. And this nystatin is used for oral thrush, but amphotericin for systemic infection. And as I told you, azoles, these are the 2 kinds of azoles. These have a severe side effects, so you don't often prescribe it. It can inhibit testosterone synthesis. It can give liver toxicity, that is hepatotoxicity, GI problems, stomach issues, fever, and chills. So if we thought antivirals have high toxicity, antifungals have even worse toxicities. How about the resistance? Of course, polyenes resistant, the fungi has acquired efflux pumps. We don't know how they become resistant to azole. They do become resistant. And echinocandins, remember, it attacks the cell wall. So what does the fungus do? The fungus decided to increase its cell wall thickness. So this antifungal, the echinocandins cannot work. And the way they do it is they increase the chitin content, and they also stop growing fast. Like, remember, you stop the growth and do the fermentation. And so echinocandins can't kill this fungi. Again, like these antifungals are very few, and is what we know as of now. Again, one of the reasons why we have so few antifungals, it was harder to study fungi, right? Because it's multicellular. When something is harder to study, very few people get into that. And also fungal infections may be rampant, but it's not killing people. A lot of people live with fungal infection. That's sort of a reason for slowing down, the slowness in developing new drugs. So when we talk about antiparasites, we usually focus on Plasmodium, which causes the malaria, and malaria is a worldwide problem. And you often know it as a malaria, and now you should know that there are 5 different kinds of species that cause malaria: Falciparum, Vivax, Ovale, malaria, and Knowlesi. And you can see that Falciparum is mostly in Africa. Vivax and ovale is in Latin America and some in Africa, and ovale is sub-Saharan Africa and West Africa, and malaria is worldwide, and then knowledge is Southeast Asia. Does that mean, does it simply stay in Southeast Asia? No. As people travel, they bring it along with them, right? So all these viruses and parasites and bacteria, they have a free ride with the plane rides, right? They all take a flight to wherever the people go, and they don't pay any price for their ticket, right? And then there are, just like viruses that go dormant or latent, there are some Plasmodium species that will go dormant in liver, and this can persist for a long time and can get activated and cause problem. And this one is malaria has nephrotic syndrome. So what are the malaria treatment? The major choice of drug is chloroquine. And there is obviously problem with chloroquine resistance now. We use mefloquine, and then we can also use atovaquone and a proguanil. This is basically an artemisinin-based drug. And that was, if you guys know, maybe Fatuma knows, there was a Chinese lady who found artemisinin and many years ago, and she got a Nobel Prize a couple of years ago for that. For Vivax and ovale, if you remember, Vivax and ovale is in only certain parts of the world, you use primaquine. And these are the one they have the latent hypnocytes, right? They go into liver. So you give them primaquine, you can kill the hypnocytes. In life-threatening situations, you can, of course, give them intravenous. This is basically a summary of various options depending on which Plasmodium species. And you should often give combination. If it's resistant, you switch. A mefloquine, as I told you, is quite new. It's only introduced in 2018. And then falciparum, you do chloroquine. All of them use chloroquine. But if a chloroquine resistant, you switch. And here's a map of chloroquine resistance malaria. As you can see, it's pretty much across the malaria belt. All the countries have chloroquine resistance. And then in some cases, it's a chloroquine and sulfoxide primethamine resistance. So you have to know what is circulating, because identifying resistance is not easy for parasites. But it's, if you know there is this area, there is a chloroquine resistance. For example, in the case of Peru, you don't give them chloroquine as a treatment of choice, right? So this is something, this is WHO constantly updates this information. So if you are in the field, you should constantly update yourself where the resistance comes from. So here are the various protozoans. Those are all the parasites, these are all unicellular parasites, Giardia lamblia. And in many of them, you also see supportive therapy, right? We don't, like for example, Cryptosporidium, it causes severe diarrhea. And whenever there is a severe diarrhea, you just give them water, right? And electrolyte. So that's what I mean by supportive therapy. And of course, metronidazole for amoeba or giardiasis, which is also a fatty diarrhea. And then there's a central nervous system infections. For those, obviously, you want to have a drug that stops them. Having said that, Naegleria fowleri can kill the patient in 9 days. And if you don't know Naegleria fowleri, and if you don't predict it, it's too late to save the patient. Then we have visceral infections such as cruzi and Donovani, Leishmania, and Trypanosoma cruzi is of importance to Miami because this is very common in South America and Central America, and we have a lot of people from the region, and this Trypanosoma cruzi can hide, and you can come here, you don't feel anything, you donate blood, and then if you get a blood transfusion, and in early days we didn't check. Now we do check for the presence of Trypanosoma cruzi in the blood when we get blood from people from this region. And these people can be treated. And unfortunately, there's a huge problem when you have undocumented the people in this part of the world, as well as South in California and Arizona. We do have an issue with this Chagas disease. And we don't have time to go through the Chagas disease, but it does cause major heart problems and can cause child miscarriages in women. Okay. And then we talked about Plasmodium, and then there is Babesia as well. We don't have time to talk about it. And there's also one of the major sexually transmitted diseases is caused by this parasite, Protozoa Trichomonas vaginalis. And that certainly you have a metronidazole as a treatment of choice. So that brings us to Helminths, and good. I'm at 11:15. As you can see, I'm watching the time. I told you guys it's an hour and a half. So very briefly, helminths means parasitic worms. And most worms are very visible to adult naked eye, so I don't need a microscope. But if I want to look for their eggs, I need a microscope, right? The adult forms cannot multiply, like they don't replicate like bacteria into one worm to two worms, but they can make a lot of offsprings. That's what it means by cannot multiply. There are three main groups of parasites, helminthic parasites. That is platyhelminths. There are flatworms. Within the flatworms, there are tapeworms and flukes. Tapeworms are cestodes, and the flukes are trematodes. And then those round worms are known as nematodes. And then we have these thorny-headed worms. If you guys look up online, they're quite scary looking worms with their head with a hook that can attach to your skin. And they are quite rare, but it is there. And this is mostly neglected tropical disease. Why, Fatuma? I don't know if she can hear. Yes, she's there. Why helminthic infections are mostly neglected? Fatuma, can you hear me? All right. So, yeah. I can hear you, Fatuma. All right. Joseph, why do you think these helminthic infections are mostly neglected?
I think maybe because they're harder to detect and the
I can
places
hear
that.
you.
Can you hear me now?
You can hear him?
Oh, yeah,
I am
yeah.
not hearing him.
Hello?
Yes. Okay.
Oh, I think maybe because in a lot of the places where people get infected with helmintic diseases, the treatment is not available. Usually, I think they're more common in third world and developing countries.
That's exactly right. Most of these helminthic infections happens in the third world countries along the tropical belt, right? Up in north and south of the equator between Capricorn and Cancer. And these people, and it doesn't affect the people in Caucasians or the Europe or the US, and where the most of money is spent on research and developing drugs against these microbes. And so hence it's very much a neglected area of research. Having said that, there is a renewed interest in these diseases in about last 10 years because of the movement of people from the south to the north. We have a lot more people in Mexico, from Mexico and Central and South America on the southern part of US. So there is a unit set up by Peter Hotez. If you guys know of him, if you're interested in doing a neglected tropical disease postdoc, I highly recommend go work with him. So Texas spent a lot of money building a tropical medicine institute in Houston and Galveston, and there's one in Tulane in New Orleans. Of course, though that one got decimated by Hurricane Katrina, sorry. And then there is one in very few, right? So I've been trying very hard to get one going in FIU, but it's not been a very easy one. They're not too interested, basically.
Isn't it like a lot easier just to deal with the sanitation issues, though?
That's one way to deal with it. So I'm going to talk about it in the next few slides, Joseph. So the one way to eliminate and control helm intake is deworming, right? It's amazing how deworming can actually literally get rid of these parasites, because this parasitic infection, and I don't have time to go through, affects people's ability to earn money, so kids to go to school. So if there's no education, no money, then there's poverty, right? So it is a vicious cycle of poverty with no education and no job. And so it's a huge economical concern, not just a concern of any travelers getting it. And for these regions, if we want to fight poverty, we have to fight all kinds of neglected tropical diseases. And thanks to Bill Gates and a couple of other entrepreneurs who really putting in the money in the helping these third world diseases, Carter Foundation as well as Bill Gates. Carter's focuses on onchocerciasis, but of course Bill Gates have more money to focus on multiple front. So for interestingly, deworming and preventative therapy, you only need to give them once a year. And whether it's a schistomiasis, fascioliasis, which is a fluke, taniasis, ascariasis, or filariasis, or sonchocerciasis, all of them, you can just need one drug and once a year to give everybody, right? But what happens? There's no money. How can the companies afford it? So in 2010, they had a London Declaration where a bunch of companies came together with WHO, and they said they're going to provide free drug to many of these countries to eliminate these diseases. So, and that has been ongoing. We have certainly seen a lot of improvement. As far as the US is concerned, any refugees coming from the region, we assume they are infected and they are given these drugs as a presumptive treatment. And of course, if you're a pregnant woman, they need to make sure you check. And if these drugs have a very specific side effects on their fetus, so you have to they have to look into which drug should give. But everybody else are presumptively treated from various drugs. And you may have heard of ivermectin, and there's a group in Australia showed that treating COVID-19 patients seems to improve the outcome of COVID-19. So with the limited study, a lot of people are going up and about trying to promote ivermectin as the wonder drug, which I don't think is the right thing to do. But most important is also vector control. Almost all these worms do have a vector, almost all, and they either snail or fish. So when you eat the snail, and now I like escargot, though I always concerned about parasites, but many of these snails are harvested and bred, so I'm quite okay with that. But I wouldn't eat the snail in Central or South America, right? Or if
Wouldn't the cooking, like,
some of them don't die easily, okay?
okay.
And yeah, but if you remember, I don't know if you have had escargot, they cook it out of it, right? They have lots of garlics and lots of coriander, and it's amazing. I love escargot.
Yeah.
Okay, and then and the fish and the snails are usually, they pick up. So many of these parasites have two forms. The form that uh, um, they call the Sylvan cycle. One is the form they live in the environment. In fact, they have three forms: one in the environment, one in the host, and one in humans. So in order to attack them, there's a different ways to attack them, because they physiologically different, they structurally different, the form is different, and I don't have time to go through it. If you're interested, you should look it up. And for example, tiniasis can cause major uh, uh, problem with your brains. Okay, cryptopsychosis, I think it's called, and there's a lot of people with that kind of huge cause of death in the Central and South America, and that comes from eating pigs and cow meat. And guess what? A lot of people like their beef rare or medium rare. Have fun. You also can acquire teniasis, right? So this is why, most Asians don't eat rare or medium rare. They want well cooked meat. So I would not recommend rare or medium rare meat anywhere because you never know. And then filariasis, mosquito is a major vector. And then onchocerciasis, onchocerciasis is a huge problem that causes blindness, and that is to the black fly that lives on the river. It's called the river blindness because black fly is found along the riverside. All right, so that's the end of my talk. And we can have a 5 minutes of discussion. Yes. So questions. Was there a lot of questions on the chat? No, just discussion. Okay, good. Well, Amrita says they do albendazole. So, we don't have time to go through this. I hope you guys, Joseph and Florida covered, and Sophia is aware of it. And that, the way you test for many of these worms is you take a swoop tube that basically take a swab of your butt in the early morning, because the worms, most of them come and lay their eggs in your butt. And then you look under the microscope, the eggs are there. And then and also it itches at night, right? Because they wiggling around your butt. And they also say that put the sellotape in your anus, and in the morning you can look under microscope and you can see the
Talking about
eggs.
pinworm?
Yeah, pinworms and other worms too, not just pinworms. And so and one time I was concerned, so I went looking for albendazole. The only place that you can buy albendazole is Navarro, which is the Navarro Pharmaceutics, only found in Miami. So people from this region, Central South America, used to getting the albendazole, and you can get albendazole. It's only a single course of treatment, right? So once you buy it, you take two cups and you're done. Not 2 cups, whatever, 2 doses.
Is there any way we can get a copy of the slides?
I could make it available for you, but certainly we are recording the presentation. Yeah. Okay. So
Do
was
you
it?
want us to
Yeah. Yes.
email
Flo.
you? Do you want us to email you those questions, or email Amrita those questions?
No, you can
Yeah.
you can put it on the chat right now. We can access the chat. So any other questions? Sophia, it's not Sophia. Fatima, are you on top of a tree? When I first came to this country 36 years ago, one of my colleagues asked me if people in Malaysia live on trees. And I'm like, what? And he says, that's what the encyclopedia said. So it looks like Fatima is climbing a tree to get the internet. Any questions, Joseph?
I think you answered all my questions like while we're doing lecture.
Okay, good. Was this a fun lecture, even though it was a quick trip through the antimicrobials?
Yeah, it was interesting too. A lot of good resources in the slides though. So that's why I kind of wanted to see me some.
Okay. All right, good. The important thing for you, Joseph and Florida, that be aware of all these things, right? You don't want to go to an interview and somebody says, have you heard of albendazole, or have you heard of this antiviral? You don't go blinking saying, no, I've not heard. But of course you have heard. If you have sat through my lecture, you've heard, right? Next semester, one of you will be assisting me with GCP program, and you will learn more in details about all this. I don't know whom I'm going to ask, but one of you will be. And even if you're not assisting me, right? Even if you're not assisting me, because Mickinson has been assigned to me in the past, he might still come back. You should attend my lecture to really learn through the whole thing, which you should have learned in the medical school, but again, it was very fast and the emphasis different. So you guys will know about it. It's twice a week, and come and attend those lectures just to sit in and get better informed. And this also might guide you in your future plans to where you want to do postdoc. And where you want to do postdoc is what area is going to be continue to be of importance in terms of research funding and research publications, right? And where you can make a difference. That's what you need to decide. And I hope Florida and Joseph know, and Samrita does know that you cannot take what you do for your PhD as a faculty member, but what you do for your postdoc, that's what it can take with you.
But I wish I could take my PhD research.
You can only take it if your mentor decides to quit.
Both of my mentors?
Anyways, all right, guys, it's 11:31. And we shall, so this is my last lecture with y'all, right? We will have one-to-one. Let me stop recording. So